Hello everyone. Good evening. Good evening. Thank you for coming again, joining me for the evening class, late evening class. We're gonna have yet another exciting lectures about animal cell culture. So far, basics molecular biology, light and electron microscope, yeah, how we do it, what are the key things we have to take care. And now is the main thing, that is animal cell culture. Yeah. How we deal with animal cell culture, because in order to work on the physiology of any disorder, it's not good to start working with the animal model straightforward. It's always good to work at the cell culture level first, and then bringing out your whole animal model. So in this way you can save a lot of money, time, and you can get very precise results. Though, while working on animal cell culture, you're working on one specific type of animal culture, not two or three cell culture. So that will be a bit, curious, because one type of cell culture might be adjoined with other cell types. So within the liver cell, if you're working on the hepatic cells, there will be other cell lines also present in the liver. So the signaling mechanism that is happening is because the all the adjoining cells, and there will be combination of signalings happening at once. So if you're working within one cell culture for one signaling, and you're finding some results that might be not true in the mouse model, yeah, so that's the difference of it. So it might be same, it might be different, then you have to prove it, bringing it on the human, on the animal model cell. So that's why animal cell culture is always given in priority. So whenever I started to work in the field of science, I always given chance to work on the first your animal cell culture, then I was given chance to work on the human or animal model. So first prove your hypothesis over the animal cell culture, then you go further at higher models. It saves a lot of time, lots of money, and makes your data even stronger, even stronger. That's the beauty of it. So slowly and slowly, students are joining. Mainly, I can see that only those students are joining me who are really wants to learn here, and the rest, I don't know, 50 or 60. I see mainly 50 students mostly present, and yeah, that's good. That's good enough. So let's just wait for 2 more minutes. Then we start with the animal cell culture. It's just 40 slides. Maybe we will finish in 45 minutes or 35 minutes. I will not take you much time, but we will make sure this topic is over today. So by end of the day, we are having a day to work also nicely done. If you want to follow me on LinkedIn, you can follow me LinkedIn for always. I will be happy to be in touch with all of you. So this is my name, Simranjeet Singh, PhD. At the moment, online professor at EduFabrica. So, so we won against Germany after 41 years. Cheers to India. Yeah, after 41 years, it's finally India's flag is shining. ठोकता German वाले ਨੂੰ बुरा. That's Punjabi stuff. uh, Pasta tear shoulder. Oh my god. Camility. Yeah, you can be part of my. Sorry, I just start scrolling. It's a very bad habit. Subscribe to YouTube channel. Mhm. My email ID you all know. MBBT animal cell culture. I think we can start now. Hi there, hi everyone. So I will shout out the name so that I can remember. I think this will be my last batch, maybe with EduFabrica. I might have some task in futures. Tanvi Lal, Soumi Mukherjee, Ashima Mishra, Aishwarya Chaudhary, Ananya Roy, Gopika Nambayar, Yogita, Sumran, Naveen, Sojit, Kunal, Dr. Urvi, Shalu, Vaishnavi, Akshay, Priyanka, Asima, Vaishnavi, Pooja. So yeah, these you are all coming on regular basis. Thank you everyone. Thank you so much for being part of this batch being session. So animal cell culture. Basically, in this, you need to understand, need to take care of your media that you're gonna use for your animal cell culture. Which kind of media, which kind of nutrients, which kind of antibiotics, which kind of supplements are required to make your animal cell culture working pretty good. Then your cell hood, how the cell hood should work, how the incubator should be, what kind of temperature, pH, carbon dioxide, all need to be maintained while you're working with the animal cell culture. Aseptic conditions, yes, has to be maintained. 75% ethanol with the gloves covered, having a lab coat over you. No cell phones in the cell culture, and just concentrate on your work. No spilling, no spreading of your media over the hood. Yeah, no mycoplasma infection of your cell culture. So that's the main idea about like how the animal cell culture works. Like, so a little bit about history. So in 1885, rocks maintain the embryonic chick cells alive in saline solution for short length of time. In 1912, Alexis Carrel cultured connective tissue and showed heart muscles tissues contractility over 2 to 3 months. The 1943 Early et al. Produced continuous red cell line, and the 1962 Bonessi et al. Published methods for maintaining differentiated cells. The 1970 Gordon Sato published a specific growth factor and media requirements for many cell types. In 79, both Stein and Sato defined a serum-free media for neural cells. And 1980 to till now, tissue culture becomes less of an experimental research field and more of a widely accepted research tool. So application of cell culture technique, then study of cell behavior without the variation that occurs in animal cell culture. So artificial organs development and in vitro propagations of viruses, vaccine development, drug effect, recombinant DNA technology, and then characteristics of cells can be maintained over the several generations, leading to good reproducibility between experiments. And cultures can be exposed to reagents like radiochemicals or drugs at defined concentrations. And we should avoid legal, moral, ethical problems of the animal experimentation. For some limitations that the standard techniques have to maintain to have healthy reproducible cells. It takes some time actually to learn aseptic techniques. For me, when I started in 2013, no, in 2011, my working with the cell culture, it took me almost 1 month, not 1 month, less than 1 month, half month maybe, 15 days, to be get used to with cell culture, because then it's become your routine. You just do your hands, you close your eyes, your hands will be moving automatically. You're splitting of cells, changing media, adding trypsin, making a new cell culture. Okay, tuck tuck tuck, this I have to do, and that I will just keep doing it on the regular basis like a robot. uh, then quantity of material is limited. De-differentiation and selections can occur, and many of the original cellular mechanisms can be lost. And cost of median maintenance of cell line has to be taken care. Then terminology, a three-dimensional culture of undisaggregated tissue retaining some or all the features of tissues in vivo. Then single cells are no longer organized as tissues, derived from dispersed cells taken from the original tissues. Then primary cell culture, derived from an explant, directly from the animal, usually only survive for finite period of time, which involves your enzymatic or mechanical disruption of the tissues. Safety has to be taken care, like there could be some cancer causing agents, cancer carcinogens, substances that can cause damage to a developing fetus, teratogen, a substance that can cause a mutation in the genetic material that can be passed to the next generation, that is mutagen. then gentamicin, thapsigargin, yeah, possible teratogens, yeah, and hygromycin, which are also one of the possible carcinogens like streptomycin, mutagen. So please take care while using them. I was using thapsigargin. Now I got to know about that it's a mutagen. In my PhD in 2016, I started to work with CTGF, connective tissue growth factor, and in that case, I was using this thapsigargin a lot, and nobody told me that it's a mutagen, and I was using on regular basis day and night. And that's the thing, while working in science, nobody tells you what are you using. Everyone at the end of the day, we are also caring for the results. Yeah, so my humble request to my dearest all fellow students here, participants who have joined me, please make sure that when you're using any reagents in your lab or any cell culture, um, make sure you read the precautions and the content over the regions, how much they have problem, how much they are carcinogenic, how much they have deadly effect on your body, on your skin, and then carefully use them. Yeah, so yeah. So sometimes you have to do radiography to check your southern blotting or some more like complex thing. The person who is going to the radiogram room, so the radio waves that the person gonna be infected with, that's are also very dangerous. So all the experiments that includes radiology, say no to that. I don't want to do that experiments. Yeah, because your life is much important than the results out of your experiments. Madame Curie, Madame Curie, who discovered the radiology, who got the Nobel Prize winner for the same. Um, she died because of the radiology actually. But she and her husband, they worked. She got two Nobel Prize winners. She was the first person and first lady, Maria Sulema Sklodowska Curie. I think it's a Polish name. Yes, it's a Polish. 1867. I mean, she's an inspirational lady, the person who are passionate about science. At just the mere age of 66, she could do a lot in her life. Yeah, she could really lot in the life in France. Cause of that, bone marrow failure, possibly. Milo. I don't want to make you afraid or make you fear here, students. I just want to make sure that in earlier time, um, aplastemia, anemia, most likely exposure to the radiation. Citizenship from Poland, France by marriage, University of Paris, yeah, Marie Curie and Pierre Curie, they were together working on this. They got Nobel Prize in Physics, David Medal, so many prizes, Albert Medal, Nobel Prize in Chemistry, Nobel Prize in Physics, yeah, Willett Gibbs Awards. Oh my God! Within this 66 years of age, within the science, they were here to do something, yeah, something remarkable, something remarkable. Beautiful, beautiful. It's good to read sometimes if you want to get inspired to be in science, want to do PhD, want to go for your masters, or want to be a medical doctor and do research side by side. Within the science, you need to be really passionate and very thorough and very like the Arjuna has this thing with the bow and arrow, and they asked what you're saying. He said, just I'm seeing only the eye of the sparrow. That's it. And then hit the bow and it's straightforwardly hit the eye of the sparrow. Yes, yes, that's the beauty of them. So yes, nowadays protective gears are mandatory, but still be careful. So that's why I just want to make sure that while doing experiments, you take care of yourself. Safety measures, use of cell culture areas. Do not bring your friends. Do not eat or drink. Or just open the Lay's chips, Uncle Chips, having a Coke. Yeah, and then yeah, 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 chilling around. Okay. and using mobile phones. So that's not the place. The animal cell culture is. These places are meant for the cafeteria, your restroom, your even in your office room you can have it, but not in the animal cell culture. We are always a lab coat, yeah, in the cell culture area. That's also some people not do. Yeah, the PIs, the professors who are higher level, they are making, they're just coming in their clothes, and they, yeah, don't tell me to wear lab coats. I've been working in my whole life, so I don't need lab coats. But it will create problems. Why? Because person coming from outside, it has dust particles and microbes which are present outside, and the cell culture lab, they are mostly tightly closed from the airly closed, so no microbe could enter into the lab. So once you enter the lab and you do not wear the lab coat, which is specially meant to be used for the lab, so this will not protect the microbes that are present on your shirts. So that's why it is always said wear your lab coats. On your hands wear gloves, and then after wearing the gloves, clean them with a 70% ethanol. Rub your hands properly. It's like a surgeon doing an operation. Just watch some YouTube videos I'm watching in late nights. A surgeon going for the operation, and then he will clean his hands with a very special soap till here, and then then the even wearing his lab coats and all the surgical stuff, his nurses are helping the main doctor in going for the surgery thing. So that's the thing. If you want to work in these atmosphere, so aseptic conditions have to be very much taken first priority. Your utensils, the cell culture setup that you want to use, your cell plates, the equipments, they all need to be autoclaved. Yeah, at 197 degrees Celsius, airtight pressure. These are all also important, very important. So yeah, wear disposable gloves, but make sure dispose of them in the correct way before you leave the area. So when you will be working in the labs, there will be two or three different kinds of dustbins. One will be white, one will be yellow, one will be blue. In the white ones you throw out plastic, in the blue ones you throw organic, all the cell culture waste, and in the yellow one you throw away the radioactive or very dangerous stuff. And do not wear disposable gloves in the corridor or write up areas, because these things I was doing actually. So while working in the lab, once I'm done with my cell culture, so with the same once I came from the lab, I'm still wearing the same gloves and I'm holding the pen and writing in my lab book. Okay, I did that today, I did that, and then my lab book is also contaminated. I made my pen contaminated, yeah, and it's all kaput, all waste. And I will make my members who will come and be part of that lab book, it will be also bad for them. So please make sure you take care of all these small things. I'm pretty sure there are 25 or 30 students here. The 20 student at least out of you will be doing animal cell culture, and things that I'm providing you at the moment. You will make sure that you also do at least 80% of that what I'm saying. 20% is okay, you can skip some things, but believe me, this will help you on the to become a good molecular biologist or as a scientist in the future. So basic equipments that are required for the animal cell culture is horizontal laminar flow cabinets. These provide the most sterile environments for the cells but offer no protection to the operator. And then filtered air enters at the back of the cabinet, which is directed to the front directly at the operator. And the most sterile part of the cabinet is at the back, which centrifuge and each cell culture, which are refrigerated. Human derived cell must be centrifuges in the sealed rotors. 100 G is hard to enough sediment cells, so higher G forces may damage cells. If tube breaks in the centrifuge, take the whole bucket into the cabinet and clean it there. So make sure you make all these things also. Contamination of cell culture that I'm keep talking about. Microorganisms could grow 10 to 50 times faster than the mammalian cells. and they take around 8 to 16 hours to divide, and they are more tolerant to the variations in the temperature, pH, and nutrient supply. They are the, I don't want to use that word, they are the bad T cells of the, yeah, of the whole animal cell culture. They makes your life vulnerable actually, that you're working with so many careful nature, but still things got bad and makes your cell culture infected. So in order to do so, it's always suggested to use antibiotic resistance microorganisms, and mycoplasma is the important cell culture contaminant. So what we used to do, twice a month, we check this mycoplasma test. So we take some media from one of the cell culture that are present in the incubator and check for mycoplasma test. If it is present, that means it will be present in all the cell culture. And these mycoplasmas, they're very tiny cells, and they're just moving so fast like a superman in the cell culture, that and they're growing so rapidly, making your cell culture feel so bad and vulnerable, and they can't do anything. They are like, I don't know, name any villain of the Bollywood films. I don't remember the villain's name. Yeah, I stopped watching Bollywood actually. I stopped watching any movies actually. I should watch some. Can you suggest me some movies I should watch maybe? Yeah, that's a joke apart. So they are the bad people, mycoplasma. They should be never be in your lab. And mostly they are coming if you're eating chocolates in the hidden corner of a cell culture, just to make yourself calm down. Even we used to eat, and then using the same hands and then going for the cell culture, and it will make that happen. So isolation of cell lines for in vitro culture, resected tissues, yeah, then shallow tissue culture in vitro, then primary culture, then you make a subculture out of it, then secondary culture, make a subculture out of it, then you have a cell line. And this cell line could go into three different parts, that is single cell isolation, immortalization, and successive subculture. From the single cell isolation, clonal cell line. From immobilizations, transform cell line, and then successive subculture senescences happen. Then you have immortalized cell line at the end. So types of cell culture in vitro. So derived directly from animal tissue. So these are primary cell culture and secondary cell culture. Primary cell culture are the best ones so far. We take them from the animal tissue, embryo or adult, normal or neoplastic, cultured either as a tissue explants or single cells. Initially heterogeneous, become overpopulated with fibroblasts. Finite lifespan in vitro, retain differentiated phenotype, mainly encourage dependent exhibit contact inhibition. Yeah. Then secondary culture, they are developed from the primary cell culture, isolated by selection or cloning, becoming a more homogeneous cell population. Finite lifespan they have, and they could retain differentiated phenotypes. Encourage dependent, and they exhibit contact inhibition. And third type is your continuous culture. They are derived from primary or secondary culture. They are immortalized spontaneously by transformation vectors, serially propagated in culture, showing an increased growth rate, homogeneous cell populations, loss of anchorage dependency, and contact inhibition. Then infinite lifespan they have in vitro, and differentiated phenotypes retained to some degree in cancer-derived cell lines, and very little retained with the transformed cell line. They are genetically unstable. Then cell morphologies depending on the cell types. So this is fibroblastic. This is epithelial cell line. This is endothelial cell line, and this is neuronal cell line. Yeah. So these are the four different cell lines. So in the fibroblastic, here the cell lines are like that. You can see the cells being separated from each other, very tiny and in elongated shape. The epithelial cells here, they are more into condensed into this network of spiders, we can say spider web, and this is more like dense and flattened cell types joined all together, the endothelium. These are the neuronal cells, shiny from the center part, neuronal cells, because of the neural activity. Yeah. So all morphology, all four different cell types, they are all different from each other. Then types of tissue culture. One is primary, one is continuous. Primary is a normal cell culture without any change in the division rate. Continuous is a finite and indefinite. Finite is a single tap roughly 30 times of division, and indefinite is nearly the same as finite, but the cells can divide indefinitely by transformation into the tumor cells, and they are called cell lines. So main types of cell culture. So we have discussed primary culture, which is we get it from the normal animal tissue. We cut part of the animal tissue, that's the primary culture. Then comes the continuous culture, which is from the continuous single cell type. Then normal diploid cells maintain some degree of differentiation, finite life. Then established cell line, the tumor cells transform cells, aneuploid number of chromosomes, and limitless availability. So this is like Morura, this is blastocyst with the inner cell mass, and this is ESC culture with the muscle cells, blood cells, neurons, intestinal cells, pancreatic islets, and liver cells. So that's the how your different cell types could be differentiated with the help of this animal stem culture. Yeah. Infinite Kanishka, it's the like there is a cancer cell line. Um, so if you want to work with any breast cancer cell line, I work with the MBA 231. So what they were crazy cell line actually. So if I put them in a media, one let's say um, 50 cells or 100 cells per micro ml per ml of the media, and within one day they will grow bunch of cells and they cover whole cell plate. uh, it's um, one of the fastest growing, and they grow infinitely. So that's the thing that, with the cancer cell line, if somebody wants to stop their growth, wants to stop, because you know what happened in the cancer, if the cancer is happening let's say in the heart or in the liver or in the kidney, then once it's become a tumor, then it tends to go transfer via blood cell line, the blood flow to the another organ. So it will have a primary site and then it will be secondary site, and then going from primary site to secondary site via the blood flow, that is known as metastasis. Because cancer cell lines, they never stop growing, they are infinite lifespan, they never stop, they are immortals. That's why they are called, like the movie 300, I remember I watched during my college days, 300. So these 300, they were tell they were immortals, nothing could happen to them, they have this god grace and so on. So um, so same way is the cancer cell lines. Once they develop in the body, and no one can stop them. That's why the chemotherapy, radiotherapy, natural therapy, all therapies a person has to go through to first of all stop at the primary site so that it could stop flowing in the another organs. So that's why there are different stages of cancers. So if it is flowing in the another organs, then this is the stage 4. That's why they say the patient is very difficult to be saved. You can either take care of that person for better health. Yeah. So that's the thing. Yeah, the same thing. So they are self-renewal cells with the pluripotent stem cells, and they could differentiate it into mesoderm, endoderm, and ectoderm. And these stem cells, the mesoderms could make RBCs, skeletal muscle cells, and cardiac muscle cells. And endoderms could make pancreatic cells, lung cells, and thyroid cells. And ectoderm includes your pigment cells, neurons, and skin cells. So that's the one of the example with the pluripotent stem cells with the hemoptysis, um, hematopoiesis. Sorry, you can have myoblast as a monoblast with the eosinophilic myelocytes, basophilic myelocytes, monocyte granulocytes, and progenitors. So they will specifically grow eosinophile, basophiles, neutrophile, and monocytes, right? And then this myoblast, monoblast will produce basophilic erythroblast, orthochromatic erythroblast, retrocyte, and erythrocyte, and so on. So this is the resting state, yeah. This is a G2 checkpoint for cell size and DNA replication, and this is a spindle assembly checkpoint, check for chromosome attachment to the spindle. And this is the G1 checkpoint for cell size, nutrients, growth factors, and DNA damage. And for the resting state is G0. Then cell culture environment. How should be the cell culture environment should look for? So substrate or liquid. So nutrients, it should contain culture media. Then environment, it should have your carbon dioxide temperature 37 degrees Celsius, and humidity has to be taken care. And oxygen tension maintained at atmospheric but can be varied. And sterility, aseptic technique, antibiotics, antibiotics, and mycoplasma tested afterwards. Then cell culture environment, so basal media which maintains pH and osmolarity, yeah, which provides nutrients and energy source. So what are the components of basal media? Inorganic salts which maintain osmolarity, regulate membrane potential like sodium, potassium and calcium. And the ions for cell attachment and enzyme cofactors. So pH indicator, phenolride is also added into that, which is the optimum cell growth. Approximately pH is 7.4. So red color is shown here with a neutral pH around 7.4, which is optimal. If it is become pinkish, it's become acidic. If it is become more reddish, it become more basic. So both things are not good for animal cell culture. If you are growing your animal cell culture, if you're seeing it's becoming yellowish, that means it become acidic and it's not good anymore. You should throw your cell culture. If it has become more reddish, that means it's more basic, and again it's not good enough. So in order to maintain that, you have to passage your cells after 2 or 3 days. You need to split your cells, add new medium, and then continue afterwards. Then buffers have to be added, bicarbonate buffered media, which requires carbon dioxide atmosphere. Then HEPA strong chemical buffer, which range pH 7.2 to 7.6. Then glucose, which requires your energy source. Then cell culture environment, the components of basal media, it includes your keto acids, including your oxaloacetates and pyruvate, and intermediate in glycolysis and Krebs cycles. Keto acids added to the media as additional energy source, which maintains maximum cell metabolism. Then carbohydrates, energy source, glucose and galactose, low and high concentrations of sugars in basal media. Then vitamins, they are the precursor for numerous cofactors, and B group vitamins necessary for cell growth and proliferation. And common vitamins found in basal media is riboflavin, thiamine and biotin. And the trace elements, it includes zinc, copper, selenium and tricarboxylic acids intermediates. And cell culture environments, which includes your supplements like L-glutamine, non-essential amino acids, growth factors, hormones, and antibiotics, antimitotics. So these are the various functions. Like L-glutamine helps for the essential, which is essential amino acids, energy source, unstable in liquid media. That's why I did as a supplement. And NEAA, non-essential amino acids, to the basic compositions, reduce the metabolic burden on the cells. Then growth factors and hormones like insulin stimulate glucose transport and utilization, uptake of amino acids, maintenance of differentiation. Then antibiotics and antibiotics to maintain from the from the bacterial and fungi infection. And also in your cell culture, you add your fetal calf bovine serum, that is FCS and FPS. So these are growth factors and hormones which aid cells attachment, binds and neutralize toxins, and has a long history of use. And disadvantage is that it's an infectious agent, variable compositions, expensive, regulatory issues are required to minimize the risk. It has heat inactivation 50 degree Celsius for 30 minutes. Yeah, why? To destruct the complement and immunoglobulins in them, and destruction of some viruses also. This is fetal bovine serum, FCS. This is also being highly used in the animal cell culture. So how do we culture in the laboratory? So here we can see containment level 2 cell culture laboratory, which has typical cell culture flask, and Mr. Frosty, which used to freeze cells. Yeah. So this is your revived frozen cell population, which isolate from tissue, maintain in cell culture, aseptic technique. Subculture is done passaging, then you count your cells, and then you can cryopreserve them. So how does the passaging of cell is done? Why it is required to do passage your cells? It basically maintains, as I discussed, the cells in culture, so that the cells should not overgrow. To increase the cell number for experiments, right? So you check the confluency, how much confluent your cells are. So here we can see the confluency is around 70 to 80%, and this is the 100% confluent cell line. So you should passage your cells here only at 70 to 80%, because this one is so much cells are there. So the biochemical pathway and everything is not good enough to work on that. And then you remove the media that is present in it, then wash with the PVS, then incubate with the trypsin to remove this cell line from bottom, and then resuspend again in the media, and then transfer to the culture flask. So let's check how this passaging is done. And believe me, if some of you will be working in the lab in the upcoming time, make sure you make your plans of your day before going home. Yeah, if you want to work tomorrow, so if you're working late night till 7, so by the 7 p.m. you make sure give your 30 minutes what you want to do tomorrow. Basically you should plan the whole week plan on Friday or on Saturday. And then then you start your lab by 8 a.m. in the morning, so that by the 10 o'clock you're finished with your all work and successfully. So by the 11 and 12 you're reading your articles, you're making other documentations, writing your paper, and doing other stuff, all the stuff. So in this way you can maintain a balance of your life. So let's check how the passaging of cell culture is done.
The growth of cells in culture follows a standard pattern. A leg after seeding is followed by a period of exponential growth called the log phase. Cells should be passaged when they cover the plate, or the cell density exceeds the capacity of the medium. Maintaining log phase growth will maximize the number of healthy cells for your experiment. Before retrieving your flasks from the incubator, sterilize the hood and have all required supplies at hand. Examine your culture carefully for signs of contamination or deterioration. Handle the cells with care during transport. For adherent cells, remove the spent medium from the flask using a sterile pipette. Rinse the cells with a balanced salt solution, such as DPPS. Make sure to use a salt solution without calcium and magnesium, as these may inhibit your cell dissociation reagent. After rinsing the cells, remove the salt solution. Each time you aspirate liquid off the cells, place the next solution on quickly. Add your cell dissociation reagent to remove cells from the plate. Use just enough solution to cover the cell sheet. Consider using a gentle dissociation reagent, such as Triple Express, to avoid damaging your cells during dissociation. Make sure that the solution completely covers the cells. You may want to tap gently on the plate to help the cells detach. Use a microscope to confirm the cells have released from the flask. They will start to appear round as they release from the substrate, and will move or slide when the flask is tilted. Complete dissociation is necessary. Do not leave the dissociation reagent on too long, especially if you're using a reagent other than triple. You may choose to manually break up lingering clumps by repeatedly pipetting warmed medium over them. A single cell suspension is important to achieve an accurate cell count. If you are using trypsin, the collection medium will need serum, or you'll need to use a trypsin inhibitor to inactivate the trypsin. If you're using triple, inactivation is achieved by dilution alone. No serum is needed. Transfer the cells to a conical tube and centrifuge to remove any residual dissociation reagent. The speed and time of centrifugation will vary based on your cell type. After centrifugation, you should have a well-formed pellet. Remove the medium from the centrifuge tube with a pipette and discard the medium into a waste container. Try not to disturb the pellet. Resuspend the pellet with warm, complete growth medium. Gentle pipetting will disperse the cells to ensure a homogeneous solution of single cells. Remove a small sample for cell counting. Trypan blue is used when counting cells to indicate the ratio of live to dead cells. The stain turns dead cells blue, but healthy cells with an intact membrane will remain white or colorless. Using the microscope, count the total number of cells and the number of dead or blue cells. Based on your cell count, determine how much additional fresh medium to add for optimal seeding density. Add the required medium, mix the cells gently, and pipette the solution into fresh flasks. Cap the vented caps on your flask tightly. If the caps are not vented, cap loosely. This keeps contamination out while allowing adequate gas exchange. Use a north-south, east-west motion to evenly distribute the cells and transport the flasks to the incubator. When passaging suspension cells, you'll begin by removing a small sample from the cell culture flask for counting. You'll follow the same counting procedure for adherent cells using Trypan Blue and either a hemocytometer or the Countess automated cell counter. Based on your cell count, add additional fresh medium to the flasks. Stay within the minimum and maximum volumes. This is important to maintain optimal air exchange and shaking flow. You may need to split the culture into multiple flasks. Cap the flasks appropriately, depending upon whether they are vented or not, and return them to the incubator. Every 3 weeks, gently spin the cells, remove the medium, and replace with completely new medium. This eliminates cell debris and metabolic waste.
So I hope this video has cleared you from any perspective. It's a horizontal one. So you can passage these cells on normal basis like this. Then cryopreservation. After passaging, you can do cryopreservation. You resuspend your cells in containing media, then centrifuge and aspirate the supernatant, then resuspend your cells in 10% DMSO in FCS. So that's the thing which will make your cells freeze. Then transfer to the vials at the -80 degrees Celsius. Transfer the liquid and nitrogen storage tank. So by doing cryopreservation of cells, you reduce the risk of microbial contaminations, reduce risk of cross-contamination with other cell lines, reduce risk of genetic drift and morphological changes, and research conducted using cells at consistent slow passage. And how we do it? With a long phase of growth, passage cells and parallel for the medium exchange. So we have seen in this movie, in the passaging of the cell, this hematocytometer, you count the cell number and then you calculate the cell count of it. There's also automated cell count, which count your cell lines automatically. So this is the ideal growth cell culture that we see. The typical one, you have a lag period, an exponential phase, then stable plateau. With the cells per ml days from subculture and cells per centimeter. Then contamination. So cell culture contaminants can be defined as some elements in the culture that is undesirable because of its possible adverse effect.
1 on the system. 2 on the system. 3 on the system. 4 on the system. 5 on the system. 6 on the system. 7 on the system. 8 on the system. 9 on the system. 10 on the system. 11 on the system. 12 on the system. 13 on the system. 14 on the system. 15 on the system. 16 on the system. 17 on the system. 18 on the system. 19 on the system. 20 on the system. 21 on the system. 22 on the system. 23 on the system. 24 on the system. 25 on the system. 26 on the system. 27 on the system. 28 on the system. 29 on the system. 30 on the system. 31 on the system. 32 on the system. 33 on the system. 34 on the system. 35 on the system. 36 on the system. 37 on the system. 38 on the system. 39 on the system. 40 on the system. 41 on the system. 42 on the system. 43 on the system. 44 on the system. 45 on the system. 46 on the system. 47 on the system
This we have discussed. This we have discussed. Some comparisons, properties of finite and continuous cell line. So finite, which is like some definite period, the growth rate is slow, the continuous is fast. This is monolayer, suspension, low and high, normal, immortal, euploid, aneuploid. Anchorage dependence, yes, no. Contact inhibition, yes, no. Cloning efficiency, low and high. Serum requirement, high and low, and so on. So these are the common cell lines that we use for animal cell culture. Human cell line, MCF7, this is the breast cancer cell line. For leukemia, HL60, HEC293, human embryonic kidney cells, HeLa, Henrietta Lacks, primate cell line, Vero, African green monkey kidney epithelial cells, COS7, and other CHO from hamster, SF9 and SF21. Now comes your organ culture. So we have discussed about animal cell culture. So there's also nowadays very much demanding with the 3D bioprinting organ culture. You can print your own organs, freshly heart or kidney. So this is the futuristic studies that we want to do. So these are grown some in small level. Your artificial organs. I think this is heart. This is kidneys, and this is, I'm not sure what's that. It's maybe something very ugly. So this is the upcoming time now. So thank you very much for your time. I will not take you much time because you might be a bit sleepy now, I guess. So I will share this video and principle of cell culture and fundamental techniques in cell culture, one PDF, and from the European Union Cell Culture Basics Handbook. So all are beneficial from the cell culture perspective. Okay, good. So we are done for today. So thank you very much, everyone. So wish you all nice evening here. And tomorrow we will start with yet another exciting lectures that is about molecular biology methods. I think this one works for us or not. Yes, this will be a lot of students. We will be doing a lot of virtual labs in that actually. So we'll be dealing with PCR that we have done a little bit about gel electrophoresis, blotting technique, real-time PCR, microarray, recombinant DNA technology, Sanger sequencing, next generation sequencing, cell culture methods for producing steady gene functions like creating this is growth hormone is added to that. Generation of transgenic mice, CRISPR Cas9, knockout mice, knock-in mice. So tomorrow will be yet another exciting day for all of you. New things to learn. Thank you very much. I'll see you tomorrow at 5 p.m. Yeah, sharp at 5. I will send the link before, and then we start our classes as usual. Yeah. So take care. Good night. Sleep well. Make yourself proud. Make your family proud. Make everyone proud. On that note, good night. Sayonara. Ciao, ciao.